G'day Groovers. This fairly new term organ reserve might end up being the most important marker of general health, particularly in aging, that we've ever come up with. In this video, let's get an overview of organ reserve, then drill a bit deeper to see what aspects of our metabolism might give an indication of how we may be able to measure our organ reserve. And to finish up, I'll suggest some action points for increasing our organ reserve. Firstly, organ reserve refers to the ability of an organ to successfully return to its original physiological state following repeated episodes of stress or injury. Or put more simply, the functional capacity of the organs to support life. Now, the word organ in this context refers collectively to our heart, liver, lungs, kidneys, and total skeletal muscle mass. But really, in terms of metabolically active tissue, it probably encompasses more than just those. When we're young, our organs have more than enough capacity for basic function. Their limits get tested, but not seriously taxed by illness, injury, and environmental toxicity. It's thought that this excess metabolic capacity could serve as an innate defense system, which might help intermediary metabolism cope with increased metabolic demand, chronic or acute stresses, and recover from injury. As we age, however, our organ reserve diminishes and gets more severely tested and taxed by these same factors, and tends not to bounce back as well as it did before. In recent years, researchers have begun trying to give perspective to organ reserve in a physiological context by expressing how many metabolic processes display excess capacity over and above what is required for normal daily function. Or in simpler terms, they're trying to provide the big picture of organ reserve by giving examples of how many small-scale processes already possess their own reserve, or extra capacity built in, so to speak. The rate of organ functional decline has been quantified to occur at a linear rate measuring between 0.5% to 1.4% per year, and all indications are it's accelerating by our fifth decade. This reality is succinctly expressed in this quote from a recent study. These observations indicate that aging is characterized by limited organ reserve. What's more, it's interesting that organ reserve and lean muscle mass appear to be linked throughout our life. Not only that, but an ongoing effort to maintain and even build muscle mass tends to improve or retain the function of vital organs as a whole. So let's poke our nose into the biochemistry a bit and look at several specific examples of excess capacity which are emerging as candidates for measures of organ reserve. Excess capacity or capability is represented in the cell cytosol by surplus production and activity of 4 of the enzymes used in glycolysis. I'm listing them here because they take way too long to say. These enzymes are present in quantities believed to be at least 5 to 10 times more than is required for normal activity. The hexose monophosphate shunt, an offshoot of the glycolytic pathway, produces excess glucose 6-phosphate dehydrogenase, allowing for greater production of NADPH, which is crucial in protecting cellular functions from oxidative damage. Excess capacity of complex 4 of the electron transport chain is understood to be several-fold over and above what is required but declines with age. The observed decline in this complex is touted as a possible measure of reduction in organ reserve. And here's a fun fact. Complex 4 of the electron transport chain is responsible for consuming more than 95% of the oxygen used in a cell. Mitochondrial DNA, or mtDNA, is observed to have a large degree of redundancy, and the inordinately large number of mtDNA per cell might be another useful measure of excess capacity. Surplus mtDNA can be viewed as carrying out an important role in reducing the accumulation of mtDNA mutations, themselves representing a negative impact on energy metabolism, which typically occurs with age. Telomeres, quite the topic for the last couple of decades, have also emerged as a candidate for measuring organ reserve. Adequate telomere length, which reduces with age, is understood to protect genetic material and suppress the triggering of replicative senescence. If we can view telomeres as protective of genome stability, protective of normal gene expression, as delaying replicative cell senescence and aberrant cell apoptosis, then quantifying telomere length could be indicative of an organ's ability to recover from injury and tolerate stressful conditions. Also exceeding basic physiological demand is sodium-potassium ATPase in the membrane of neural cells, making possible the creation of repetitive action potentials and the release of neurotransmitters. Excess neural capacity is essential for brain plasticity and cognitive function. Here's another fun fact. Sodium-potassium ATPase is a huge energy hog, consuming up to 2/3 of total cellular energy. Okay, admittedly, I've just touched on some pretty specific metabolic pathways and biological systems, but the point of going through these isn't to complicate the discussion of organ reserve, nor make it appear inaccessible to those without a deep understanding of the physiology. It's to give just a glimpse of the variety of standout elements of human biology displaying excess functional capacity that diminishes with age whilst also being measurable. Therein presenting themselves as a possible grouping of quantifiable markers whereby a collective measure of systemic capacity may be derived and viewed as indicative of a larger decline. Or simply, an ability of our bodily organs and systems to better maintain basic functions and bounce back effectively from injury and stress. The concept of organ reserve is complex and tricky to convey, so I hope I've at least gone some way to presenting a big picture of the theory, whilst also providing a little insight into microscopic cellular pathways of real consequence when viewed as a collective. Somewhat aligned to the concept of biological aging in a sense. So to finish up, here's a couple of action points we can employ to help preserve or even increase our organ reserve. 1. Cut down on foods that cause oxidative stress and systemic inflammation that consume the excess capacity of important biological pathways. This means avoiding trans fats, omega-6 fats, too much alcohol, grain-based foods, and particularly sugar and other refined carbohydrates. These nasty foods also disrupt hormone levels that are key to maintaining and building lean muscle mass and supporting healthy organ function. Foods to focus on are nutrient-dense veggies, oily fish for omega-3s, and quality red meat, all of which tend not to cause oxidative stress. 2. Lots of moderate activity coupled with short spurts of high energy output. And nothing beats weight-bearing anaerobic activity for building muscle mass while also building excess capacity in essential physiological pathways. Avoid long-term chronic cardio exercise because it raises cortisol levels, which can deplete muscle and taxes the body's organs and the immune functions that support them. So there it is, a brief overview of organ reserve. Thank you so much for watching, and if you found this information useful, then please consider clicking the like button so more people can find it. I'll see you in the next video. Have a beaut day.